Hi, this is Farron, and today we are discussing the paper, The Collaboration of Antibody and Inflammation in Clearance of Rabies Virus from the Central Nervous System, from the Journal of Virology. The rabies virus is a bullet-shaped, negative-sense, single-stranded RNA virus. Viral infection in humans is lethal in the absence of post-exposure prophylaxis, which must consist of both the rabies virus-specific DNA and active immunization against the virus. Based on the fact that upon infection, the rabies virus likely reaches the CNS prior to treatment, we consider that active immunization and rabies post-exposure prophylaxis provides an element of immunity, which is distinct from virus-neutralizing antibody. The study starts off with the basis that immune defense against viral infection of the CNS is limited by the blood-brain barrier. Viral infections of the central nervous system are contained with cooperative action of immunity, such as soluble factors, antibodies, and cytotoxic T cells. Soluble factors include interferon. Type 1 interferon have direct antiviral activity, and along with type 2, there is collaboration in the cell-mediated viral response. Interferon gamma has a major influence on cellular immunity as well. Mice lacking interferon alpha, beta, and gamma receptors are unable to mount a cytotoxic T lymphocyte response to LCMV. Similarly, neutralization of interferon gamma impairs the clearance of measles virus. CD8 T effector cells have been shown effective in reducing viral titers in the brain after several different viral infections, such as coronavirus and LCMV. Virus-neutralizing antibody also plays an essential role in virus infections of the CNS. Treatment of rabies-infected rats with the VNA protected the animals against lethal infection and cleared the virus. Virus-specific antibodies are necessary for the elimination of free virus, but it is speculated that the interaction of several immune functions are required for virus clearance from the CNS. To test this hypothesis, the chosen model system is based on infection of adult mice with the attenuated rabies virus CVS F3. By comparison to the wild type CVS24 rabies virus, which is the same antigenically, the CVS F3 variant has reduced cell-cell spread in vitro and limited replication in the CNS of immunocompetent mice due to a single mutation at amino acid 333. To delineate immune effector mechanisms involved in clearance of viral infection, as described in the last slide, the course of the CVS F3 infection and the development of immunity in gene knockout mice has been compared. The mice include CD4 and CD8 deficient mice, which are beta-2M knockout mice, and their corresponding conjugate control, C57BL6. T and B cell deficient mice are RAG2 knockout, and their corresponding conjugate control are 129SBEV. Antibody deficient mice are JHD knockout. These mice, along with C3-C4 complement knockout mice, are both on a C57BL6 background. We are also looking at interferon alpha-beta knockout as well as interferon gamma knockout mice, which are both on a 129 SBEV background as well. To look at the development of clinical signs in normal and gene knockout mice infected with the virus, the mice were infected intranasally. Intranasal infection was used as it provides ready access to the CNS while minimizing any local immune response. The mice were then examined for clinical signs of disease and weighed daily. The normal mice lost weight peaking at day 11 and 12, then regained their weight around day 21. RAG2 and JHD knockout mice lost weight progressively and eventually succumbed to the infection around day 21 and 24. All other knockout mice showed no significant difference to that of the immunologically intact normal mice. To look at the clearance of infectious virus from the brain at different times post-infection, the mice brains were removed. A 20% brain suspension in PBS was prepared. Virus yield was then determined by infecting mouse neuroblastoma cells with brain suspension at serial tenfold dilutions. Cells were then fixed and subjected to fluorescence staining and counted using a fluorescence microscope. The virus was present in all mice at day 8. At day 13 and 21, infectious virus only remained in RAG2 and JHD knockout mice. The titers of the RAG2 knockout mice increased roughly tenfold, while JHD knockout titers remained constant. The mice unable to make antibody died between day 21 and 24. The findings argue that there is a contribution to the antiviral response from the T cells in addition to their important role in helping antibody production. The development of neutralizing antibody response may interfere with the isolation of infectious virus from the brain, which could lead to the false perception that the virus had actually been eliminated. To control for this, we examined the clearance of viral RNA and viral antigen. Total RNA was isolated from the brains of the infected mice, and reverse transcription reactions were performed. Rabies virus-specific PCR products were blotted onto a membrane. At day 8, rabies virus was present in all brains. At day 13, viral RNA was cleared from the normal and C3-C4 complement knockout mouse brains. This recovery pattern is consistent with the hypothesis that antibody-mediated clearance from the CNS is not dependent on the participation of better-known complement cascades, although that does not rule out the contribution of component C1. At day 21, viral RNA was still detected in RAG2, JHD, beta-2M, interferon alpha-beta, and interferon gamma knockout mice. Any mice surviving 37 days no longer had viral RNA detected. Since the virus is cleared in the absence of functional CD8 cells, this indicates that the cells are not necessary for the clearance of the virus, although it does seem to accelerate the elimination of the virus, perhaps through the production of soluble factors. The differences in the rate of clearance of viral RNA are supported by the results of immunohistochemical analysis for rabies virus N protein in the brain. At different times post-infection, mice were anesthetized and perfused transcardially with a PBS solution and a fixative solution. When the brains were removed, they were post-fixed in the same fixative solution. The sections were then stained with antibodies specific for rabies virus N protein. N protein is detected in all the brain sections at day 8 and 13. At day 21, the N protein was not found in normal mice and complement knockout mice. The N protein was present at low magnification in RAG2 knockout, JHD knockout, beta-2M knockout, and interferon gamma knockout mice. The presence of rabies antigen-positive neurons were shown at day 21 in RAG2, JHD, beta-2M, and interferon gamma knockout mice. And as you can see, there is clearly a difference between these and the normal mice, shown in E and F. Look at the production of virus-specific antibodies after injection with rabies virus. 10 and 20-day blood was collected and heat inactivated. The isotypes of rabies virus-specific antibodies was assessed using ELISA. Table 2 shows that RAG2 and JHD knockout mice cannot make virus-neutralizing antibody, and thus they did not clear the infection. All mice except for these tested high in VNA titers by 20 days post-infection. Since clearance of rabies virus from the brain is dependent on the presence of VNA, it is not surprising that RAG2 and JHD knockout mice, which cannot make VNA, eventually succumb to an infection. The interferon alpha-beta and interferon gamma knockout mice, VNA production was delayed. Beta-2M knockout mice produced titers higher than the control, even though they failed to clear the infection by day 13, as previously shown. Therefore, in addition to the quantity of VNA, the quality could also play a role. It is possible that differences in local cellular response are also involved in the clearance of the virus. This is reflected in the level of cell infiltration into the CNS. To look into this possibility, coronal sections were prepared and stained to identify the presence of these infiltrates, which are usually the highest in the hippocampal fissure. Normal mice at day 8 and 13 showed massive cell infiltration, which was nearly resolved by day 21, resembling the uninfected controls. In beta-2M knockout mice, there was a significant inflammatory reaction, but it was delayed. In RAG2, JHD, and interferon gamma knockout mice, there was no inflammatory process at day 8 and little to no inflammatory process at day 13 and 21. These studies confirm that rabies viral neutralizing antibody is a requirement for clearance of the established rabies virus infection, as mice lacking B cells do not clear the virus and succumb to the infection. Production of antibodies alone, regardless of isotype, was not sufficient for rapid elimination of the virus. Although, for clearance of the virus to occur, there must be a collaboration between VNA and inflammatory mechanisms, as a strong local CNS inflammatory response may promote contact between immune effectors and infected cells, or enhance the antibody production more directly through factor production. An intact interferon alpha-beta and interferon gamma-dependent inflammatory pathway appears to be necessary along with virus-specific antibodies to likely have clearance of the infection in human post-exposure treatment situations.